Hi everyone, welcome back. Today's presentation is about genetics, and everything is mostly from Campbell's biology. Today we'll go over Mendelian, non-Mendelian genetics and Hardy-Weinberg equilibrium, as well as Hamilton's rule. First, Mendelian genetics. So the background on Mendelian genetics is this monk named Mendel bred pea plants in a monastery around 1857. And there's a reason why pea plants were the exact right plant for the job. First reason was that there were different varieties, like white or purple. And some vocabulary to know is that a character is something that can be inherited, such as flower color, whereas a trait is a version of that character, so whether the flower is purple or white. Secondly, another reason why pea plants were good for the job is that their breeding could be controlled. So essentially what you could do was remove the pollen manually and to the carpels, and this way you can avoid the flowers self-breeding. In some vocabulary, true breeding is a plant that makes the same offspring through many generations by self-breeding. Hybridization is when you cross two true breeding organisms. Lastly, pea plants have short generation times and they have a lot of offspring. So some vocabulary to know here is the P generation is the true breeding parents, so P for parents. And then the F1 generation is hybrid offspring. So the reason it's called F1 is F stands for filial. So yeah, the F1 generation is the offspring that comes from the P generation. And then the F2 generation is the offspring from the F1 generation. So some other vocabulary to know. A gene is a sequence of nucleotides that's passed on to parents to offspring. An allele is an alternative version of a gene. And essentially where this comes from is that the DNA at the gene's locus, which is like where it's located, is different. So these small changes can cause different changes in appearance. For example, a yellow pea versus a green pea. And generally in Mendelian genetics, an organism gets one allele from each parent for each gene. Now, a dominant allele is the one that determines the appearance, whereas a recessive allele doesn't visibly affect the appearance. This will be important later. Homozygous is when the two alleles you receive are the same, and heterozygous is when the two alleles are different. And this heterozygous plants or organisms can't be true breeding. A phenotype is the observable traits of an organism, whereas the genotype is the genetic makeup, so the alleles. Okay, so there's two big laws that Mendel came up with when he was doing his research. So the first one is the law of segregation. So the law of segregation is when the alleles segregate in meiosis and they go into different gametes. So if you can see here, on the left, the blue chromosome goes into the cell on the left, whereas the red chromosome goes to the cell on the right, and ultimately the cells end up with different chromosomes that are half of what the parent cell had. Next, the law of independent assortment says the way one allele pair segregates doesn't affect the way other allele pairs segregate. So if you see here, we start off with two P's. One is capital S, capital Y, lowercase s, lowercase y. Now, if you see, the way that the S's separate doesn't really have much to do with the way the Y segregates, right? So we can have capital S, capital Y, capital S, lowercase y, lowercase s, uppercase y, and lowercase for both. Okay, and a really useful tool that's used in Mendelian genetics to predict offspring genotypes is the Punnett square. So first is the monohybrid cross, and this is when you cross two heterozygotes, but only for one gene. Now, this is technically only for heterozygotes, but now the term monohybrid cross is used for any cross of just one gene with two alleles. And same for dihybrid cross. It's where you cross organisms that are heterozygous for two genes. But it's also loosely used to refer to when you just have when you're just looking at two genes. Okay, so here's some math that has to do with probability that could save you a little bit of time. So the multiplication rule is P of A and B, which is the probability of A and B, is the probability of A times the probability of B. So here we have a dihybrid cross, and what we can see is that for color, for texture, you can find the probabilities of a certain thing in each of them and then multiply them together to get your result. So here we're looking for the probability that the offspring are heterozygotes. So if we do a Punnett square for the B gene, so for color, we see that half of the offspring are predicted to be heterozygous. So the probability of heterozygous for the first gene is 1/2. We do the same for the second gene, and the probability that the offspring is heterozygous is 1/2 again. So we multiply those two, and we get that the probability that the offspring are heterozygous at both genes is 1/4. And the addition rule is that if you're looking for the probability of A or B, then it's the probability of A plus the probability of B minus the probability of A and B. So here, we can see that it's asking for the probability that all 3 children in the family will be the same gender. So here we have the girls and the boys. So the probability that all the children are going to be girls is 1/2 chance there's going to be girls, 1/2 chance it's going to be another girl, and 1/2 chance it's going to be another girl, and that's 1/8. We do the same with boys, and we get another 1/8. And we add those two together, and then we get that there is 1/4. And there's no overlap between this, so we don't have to subtract the probability of A and B. So we just get that the probability that they are the same gender to be 1/4. Lastly is pedigrees. So pedigrees are a way for you to look at inheritance. So you don't need to know too much about these symbols, but just know mainly that squares are male, circles are female. These are what different types of twins look like, and then this is a marriage, and then these are the children. And if the individual is affected by something, then they will be colored in. Also, the way you identify individuals on a pedigree is you use this number and you follow it by one of these numbers. So this individual is Roman numeral 2, 3. Okay, and there are tricks to seeing how inheritance works. So usually you're going to be shown a pedigree and be asked, what type of inheritance are you seeing here? And generally, autosomal dominance never skips a generation, and X-linked inheritance tends to be more common in males. All right, and now we're on to non-Mendelian genetics. Okay, so the first thing I'll explain is how sex chromosomes work. So this is how sex is determined in organisms, and in humans, females have 2 X chromosomes and males have 1 X and Y chromosome. So what this results in is that human males are more susceptible to X-linked recessive diseases. And here's why. So if you look at the inheritance of a female, what happens is that they get 2 X chromosomes. And the X chromosomes in humans is much more larger than the Y chromosome, and it's the one that has most of the genetic information. So, and the human male only has 1 X chromosome. And basically for X-linked recessive diseases, human females inherit them similarly to autosomal recessive diseases, where it takes 2 recessive alleles to show the phenotype. However, for males, essentially that 1 X chromosome determines everything. So because the Y chromosome is so small. So whatever they get on the X chromosome, whether it be the dominant or recessive trait, is what they're getting. And here is the sex determination systems for other organisms. So for a lot of insects, like grasshoppers, what they have is males have 1 X chromosome, females have 2 X. For birds, especially chickens, females have Z chromosome and a W chromosome. Males have a Z and a Z. And then for bees and I believe some other related insects, females have diploid and males are haploid. So males have half of the genetic information. Next is incomplete dominance. So this is when both alleles are expressed partially in the heterozygote. And an example is snapdragons. So they look like this. So snapdragons, they have an allele for red flower color and one for white flower color. However, when you get in heterozygote, it doesn't happen that it's either red or white. What happens is that it's both. It's a blend. And so when you mix red and white, you get pink. So yeah, that's where pink snapdragons come from. Next is codominance, which is often confused with incomplete. So here, both alleles are dominant in the heterozygote, and they're both shown. So it's not like red and white mixed to make pink. It's red and white mixed to make, say, stripes or spots. So an example is rowan cows, which are these cows. So we see red and white make red with white spots. Next, something is pleiotropy. So this is when one gene controls multiple phenotypes. So an example is pea flowers and seed coats are both controlled by the same gene. So in this example, we see that this one gene controls trait A, trait B, and trait C. So it's definitely not as simple as Mendel imagined it. That's the point of non-Mendelian genetics. It's the things that he couldn't see with his examples. And there's also epistasis, which is when one gene influences the expression of another. So an example is mice coat color. So if you see here, the A gene determines what color pigment that the mouse is getting. However, the C gene determines whether that pigment is deposited in the mouse's coat. So when the C gene, like when you get the recessive allele for that, when you get both recessive alleles, then the mice are albino because the pigment can't be deposited in their coats. It doesn't matter whether, it shows that the pigment should be black or whether it should be another color. It doesn't matter because this second gene determines that it can't be deposited in the coat. And other than this, the other ones are inherited normally when the C gene determines that the pigment can be deposited. And then polygenic inheritance. So this is for quantitative characteristics which are on a gradient. So this can be height, this can be skin color, this could be eye color, right? There is no, it's not true that, people are either, say, 4'11" or 5'8", because it happens on a gradient. So essentially what happens is all dominant gives you the tallest height, and then with every recessive, it gets shorter. So these are kind of the genotypes that are important to know. Next is Hardy-Weinberg, which is an equilibrium plus an equation that hinges on the idea that the genotypes frequencies in a population stay the same if there isn't evolution. And there are specific requirements for these, but you won't really need to know about them until we get to evolution. For now, it's going to be very clear whether you should or shouldn't apply Hardy-Weinberg. Okay, so here are the equations associated with Hardy-Weinberg. So what we start out with is a pretty intuitive one. P plus Q equals 1. P is the frequency of the dominant allele, and Q is the frequency of the recessive allele. And it makes sense that they add up to 1 because everyone is going to be either a big A or small a. So it makes sense that the frequencies would add up to 1. Now if we square both sides, what we get is this equation. And here the numbers mean something a little bit different. So P squared is the frequency of homozygous dominant individuals. 2PQ is the frequency of heterozygous individuals. And Q squared is the frequency of homozygous recessive individuals. So we can use this basically, if we have 4 of these items, we can find the 5th using these two equations. Okay, So here's a practice problem to do. So I advise you to pause the video right now and try it out because, this is from the 2013 USABO test. So it's very likely that there are going to be problems like this that show up again. Okay, so hopefully you've done the problem already. So we're going to look at it. So it says that only Illinois is known for its white squirrel population. In this squirrel population, assume that 10% of the genes are for albino coat color and 90% are for gray coat. And they're telling us that Hardy-Weinberg assumptions are true, so we can use Hardy-Weinberg to figure out what percentage of the squirrels are heterozygous. So it asks for the percentage that is heterozygous, so that means we're solving for 2pq in our equation. So based on what they tell us, we know that p equals 0.9 because it says that 90% of the alleles are of the genes are for the gray coat. And then similarly, 10% are for albino coat color. So we can assume that this 90% is for the dominant because there is more. Okay, so the easy thing to do would just be to multiply them together and then multiply by 2, and we get 0.18. And that is 18%. So that's our answer. And there's a longer way to do it, and the reason I'm showing this is just so you can see how all the equations are connected. So we come up with our big equation, p squared plus 2pq plus q squared equals 1. We square these, p squared equals 0.81, and then q squared equals 0.01. We plug it back in the equation, and when we solve, we get that 2pq is also 0.18, but 18%. Okay, so next up is Hamilton's rule. So this involves math as well. So here is the big equation. So it's about altruism, which is basically in nature when you do favors for another organism and it hurts you in some way, so you're making a sacrifice. So this is saying that natural selection favors altruism if the relatedness times the benefit to the recipient is greater than the cost to the altruist. So I'll show how you can find each of them and use it to solve some problems. So in terms of relatedness, it's good to just remember some of these. You can calculate it with a pedigree or a family tree, but I think it's just easier to remember them. So if you see parents, the relationship of parents to offsprings or siblings to each other is 1/2. The relationship of half siblings is 1/4, as well as for uncles and aunts, nieces and nephews. And for first cousins, it is 1/8, and second cousins is 1/16. Okay, so that's R. Next, for calculating B and C, B is the number of surviving offspring. That would, the number of offspring that would survive if you saved this organism. However, C is the number of offspring cost. So to you, if you save the other organism and you die in the process, how many offspring would be cost in the process? Say if you were planning to have 5 kids or if you were planning to have 2, that would be C. Okay, so here is the practice. So again, pause the video, take a look at it. This is from the 2016 Opens test. Okay, hopefully you've done the problem already, so we're going to take a look at it. So the first thing it asks for is the coefficient of relatedness between person A and person B. So it says that they are cousins, so we're just going to assume that they're first cousins because they didn't specify. So if you'll remember from that chart, the coefficient of relatedness for cousins is 1/8. So if we can look at the answer choices and we can eliminate 3 of them. So that's A, C, and D, gone. So right now we're just choosing between whether person A should save person B or whether person A should not. So we can use Hamilton's rule to do this. So we know that if RB is greater than C, then yes, person A should save person B. So when we plug our numbers into the equation, for R, coefficient of relatedness, we have 1/8. And for the number of offspring, it's pretty complicated, but not really. So it says that person B intends to have 9 children, so we put 9. However, it says that person A only has a 40% chance of saving person B. So we need to slide that percentage in as well, because if you think about it, there's only a 40% chance there's going to be 9 children. So we put that number here as well. And when we calculate this out, we see that this isn't true. And this indicates that person A should not try to save person B. All right. And thank you so much. I hope this lesson was helpful. And please subscribe for more content like this.